Okay, let's get started. It's my pleasure to introduce Danny Diaz, who is an unusual figure. He has got his PhD in chemistry here at UT Austin, but also I found out, as I meet people all over campus and what types of AI and machine learning they're doing, that he could also he was okay at coding up models. He was not bad, and so we thought we should get him to work with the Center for Generative AI and with IFML, and try and build a team of people who could really combine forces in terms of cutting edge AI, and also protein design and engineering. I was being facetious. Danny's actually really excellent at training models, so and it's really unusual to find someone like that. His title right now is research scientist. He's leading kind of the deep proteins group. The group that we sort of founded, which really flailed for about the first year as we were trying to understand, how AI could work with chemistry and biology. And after a year of sort of having meetings that were going nowhere, I think we finally caught our stride and started doing, in my opinion, some pretty impactful and interesting work. Danny's going to talk about the latest work that we've been doing. Diffusion models and generative models are very important for images, and we've been working on that for proteins. So, thanks for being here, Danny, and take it away.
Thank you for that intro. Can you all hear me? Hello? In the back? Awesome. So, thanks for that intro, Adam. My name is Danny Diaz, and today I'm going to tell you guys about a project that we have been working on, I would say, since mid to late October. This is our first public disclosure of this project. It's still very under heavy development, and a few things I probably won't get into the details of it yet because we haven't published it, but I'm just really excited to share with you guys how we've been using diffusion and generative AI for protein design. So today I'm going to talk to you guys about how we're learning the protein structure landscape with ambient diffusion. So most of you guys probably aren't chemists or biologists and aren't really sure when I talk about proteins what I mean by proteins. I don't mean like muscle milk and like what you take after you work out. I'm talking about the proteins that make life possible. So this is an AI conference, so the easiest way to explain that is that proteins are like biology's GPUs. They do all the work. So they are made up of these 20 amino acids. These are like the raw bits, or you can think of like the bricks that build the house. And it's how you combine these 20 amino acids into like a linear sequence that actually will give you so here's the linear sequence. And that based on the actual order of these 20 amino acids, they will fold into a unique 3D structure. So when I'm talking about structure, I mean like the XYZ coordinates of all the atoms in this folded amino acid sequence. And then once you generate specific structures, based on the structure, they'll have a specific function, which kind of makes sense in like the real world, right? Like the reason why cars move is because wheels are round, right? The shape dictates its function, similar to how proteins work. So, you can see, for example, here's this protein. It looks like a pore. You can kind of think of it as like a cheese grater where it allows things to pass through. Here's this one of my favorite proteins. This is called ATP synthase. It's the reason why we breathe oxygen. And it kind of works like a dam where as electrons flow down a chemical gradient, the protein spins to generate electricity. Well, this is how we generate energy in our body. Again, right, this intricate relationship between the sequence of a protein, the structure that it takes, and then the function that it executes. So today I'm going to talk to you guys about de novo protein design and how we can do de novo protein in the structure landscape. So what is de novo protein design? De novo proteins is making proteins from scratch. So like to date, we get all our proteins from nature. We find it in some insect, some bacteria, some person who's like immune to COVID, boom, we get their antibodies out of them, make therapeutics, that's how we make vaccines. So it's a lot of it's more protein engineering, where we find something that nature gives us, and then we optimize it and turn it into biotechnology. Well, with AI, we're actually working on what if we just think of a protein or a function that we want. So we're trying to inverting this, right? Usually we find a protein sequence in nature, we study the structure to optimize its function. Well, what if we can describe a function that we want, come up with a structure that can fulfill that function, and then generate DNA sequences or protein sequences and validate that we can actually execute that function. So that's what I mean by de novo protein design. And in this space, so far though, we're limited by the structures that are available in nature. So how do we generate novel structures that do not exist, that can do novel things that nature didn't think of yet? The other thing is, in de novo protein design, is that evolution does not optimize structures and their attributes for what humans want to do with them. So this is kind of another thing that de novo protein design enables us to do, is just start from the ground up. And by these attributes, I mean, how soluble is the protein? Like, you don't want it crashing out, while it's sitting on the shelf. How stable is it? Can we put it in a truck and ship it from Los Angeles to New York and it doesn't fall apart? Is it easy to produce? Does it cost us, $1,000 a gram to make or $1 a gram to make? And also, is it immunogenic? So if I put it in your arm, do you break out into hives, or are you able to actually accept the treatment or use the protein therapeutically? So it's all about making new to nature protein structures and functions. So a little bit of just structural details and jargon to talk about protein structures in the context of diffusion. Proteins have 4 atoms in their backbone. It's this, they have I can't really see. They have like a C alpha followed with a C next to it and then a nitrogen next to it. And then next to this C, there'll be like this oxygen. We primarily are going to focus on the backbone, which is just these 3 atoms surrounding a C alpha. So you can see we have C alpha next to a nitrogen next to this carbon, and we're going to kind of ignore this oxygen. But what we're going to use to do diffusion is we're just going to use the XYZ coordinates of the C alpha. Every amino acid in the protein has one. And then you can generate like a frame, which is kind of like a little triangle from this N C alpha C. You can imagine this triangle has like an orientation in 3D space. So every residue, we're just going to use its location of the C alpha and the orientation of that triangle of the backbone. And this is where we're going to do diffusion on. So we're going to perturb, add noise, and rotate the frame and move the points in 3D space arbitrarily. And that's how the diffusion is going to work. So next is, what does the de novo protein design workflow kind of look like? So we're going to generate these backbones, which is, again, just like a sequence of these triangles centered on a C alpha like this. So this would be only the backbone, ignoring the side chain, which is what makes every amino acid unique. And then once we generate these backbones, which is where we're going to be doing our diffusion, we're going to then use another AI model called Protein NPNN to do sequence design. So this will take these 3D coordinates and frames, generate protein sequences, which then we can use protein structure folding tools like AlphaFold or ESM Fold and generate a full atom protein that will hopefully have the same scaffold or 3D shape as this backbone. And then usually this is where we do some computational filtering to see how well these work together before we would go to the lab and actually buy the DNA and make them. So today's talk is going to focus on applying diffusion for backbone generation. And we're going to discuss how we come up with some computational filtering pipelines or evaluation pipelines. So how do we actually know if we're making progress on making better backbones, right? How do we gauge progress without going to the lab and actually making them, but just on the computer? Yes?
Just a clarification question. So you are not actually choosing any discrete vocabulary, because the basic structure is the same, you're just figuring out how it's oriented, nothing else.
Yeah, we don't make the discrete vocabulary while we train or generate backbones. That happens here in the eval pipeline on the sequence design side. And I'm going to go a little more details into this next. So here is a more thorough look at how, just same thing as the previous slide, but actually what we're actually doing. So we will output some design scaffold or structure from a diffusion model. This is made completely from scratch. We just start with random noise Think of like a black and white image, but of proteins. And we're going to then pass it to an inverse folding model, which will actually demask the sequence that will fit into this scaffold, so we could generate letters at each position of the string. Once we have this sequence, we can pass it to AI structure prediction tools that will actually give us a structure. And then from those, we can evaluate how good is that protein. So we can take the output protein, align it with the input backbone, and look at the error, or the RMSD, between the input and the output. And then we can also use this thing called PLDDT, which is what these AI folding tools, well, they tell you how confident they are at how well they folded the protein at each position of the protein. And then we can just average it across the protein to get a global PLDDT score. So you look at the confidence of the AI model and how well this predicted AI full protein model aligns with the input scaffold generated with diffusion. So that would be the RMSD and the PLDDT. And the big two metrics we will use here is what we call designability. And designability is just this upper left quadrant. So is the RMSD low enough? We use a 2 angstrom RMSD cutoff. And is the PLDDT high enough? And we tend to use 70 as our PLDDT threshold. So if any proteins where their PLDDT is above 70 and their RMSD is below 2 angstroms, we consider designable. And for that subset of designable proteins, we will then measure how diverse they are. Is the model just outputting the same backbone, giving us the same exact 3D structure, or what is the diversity in the 3D structures that it's generating? And the way we measure diversity is we will cluster all the 3D proteins that we generate using some clustering thresholds. We don't have to get into that. And then divide it by the number of designable proteins. And that's what we call diversity. So I'm going to keep referring to designability and diversity as our key metrics for improving the models as we do experiments, and as I walk you through how the field has evolved. Okay, so now that we know how we're going to evaluate these models and the metrics that we're going to be evaluating them with, let me walk you through how diffusion, the field of diffusion in proteins. So this field kind of really started with RF diffusion, which was the first de novo protein structure backbone model that used diffusion. And again, right, we start off with atoms that you randomly sample a 3D Gaussian in 3D space. These are just atoms randomly distributed in 3D. And we're going to iteratively remove noise from them until we finally end up with a protein. So here's like a more thorough look at how this works. We start off with random noise. The model predicts this. Then we kind of interpolate between the two. Now we're here. The model predicts this. We interpolate between the two. Now we're here, the model predicts this, and we just do this step by step by step until we end up with an actual real looking protein. And here's kind of like a video animation of this. So we start off with random atoms in 3D space, and they're going to slowly become denoised until we have an actual protein. And this looks really pretty. It makes really pretty figures and graphics. But it actually won the Nobel Prize in chemistry here in October. And it wasn't for making these really pretty photos and videos that he won the Nobel Prize for, but it's actually because David Baker took it a step further. And he actually showed that you can use these models to do things in the real world. So the first thing he did was he showed that you can make symmetric oligomers. So this is where multiple copies of the same protein come together to execute a job. So you can see here this protein has been 3 copies of it are used, and you make a C3 oligomer where they're kind of C3 symmetry. And he shows that you can do this for multiple symmetries. So you can see here's these proteins. He oligomerizes them to make these symmetric circular-like structures. And then here is what these structure prediction tools predicted. So this is like the diffusion backbone that came out on this column. And then here is what the actual full atom structure looks like. And they're pretty well aligned. But this is just all on the computer. He then took it a step further and then actually made them in the lab and then solved their 3D structure. and showed that, yes, indeed, these protein sequences that were made with AI actually give us the 3D structure that the computer is telling us they make. Then he looked at binding targets. This is very important for therapeutics, especially antibodies, which we'll talk a little bit more about later. So essentially, he shows that you can condition the diffusion on another protein and generate one that will bind that protein. So you can see here in gray are the computational designs, and in blue are the actual predicted binders. And you can see they overlap really well. And this is actual binding data of these de novo proteins binding a variety of different targets. He also showed that you can do what's known as motif scaffolding, which we'll talk a little bit more in detail later. And what motif scaffolding is, oh, sorry, I jumped ahead. This is another binding design where he shows that you can do this for making binders to the spike protein of the flu. So this is the de novo protein in blue, and then here's the flu spike protein. And then here's motif scaffolding. So motif scaffolding is we have this small little motif, so it's just like a little small section of a protein. I want to make a much bigger protein that includes this in it. So, for example, we have this small motif, so we have these 4 histidine residues that want to bind a nickel atom, but I want to put this in some brand new protein, but I want it to bind nickel. So you can see we take this motif, generate a large protein structure that has this nickel binding site in the middle. And he did it 4 times and showed experimentally that he was able to make them. Here's the solved structures. And he shows that they indeed bind nickel, which is these plots going down here, and then this one going up. It's really cool here because you see how this one goes down, but these two go up? He showed that you can make it where it binds it in an endothermic and exothermic reaction. So either it releases heat upon binding or absorbs heat to bind. And you can see this motif, he scaffolded into 4 very different looking proteins. So the whole thing, they're not duplicates of each other, but they all have this zinc and this nickel binding in the center. So hopefully I've convinced you that RF diffusion is able to do a lot of really cool de novo protein design. And you probably are thinking like, wow, what can RF diffusion not do? So this is kind of where the field is today and trying to improve it and do new things. So RF diffusion is not perfect. So here is kind of looking at the secondary structure of a protein, and RF diffusion struggles with diversity of secondary structure. So what I mean by that is proteins tend to have these helices and these beta sheets, which are these arrows that point as they go, and they can be parallel or anti-parallel. And they also have these loops, these gray parts that connect these secondary structure elements. And RF diffusion tends to struggle with making proteins that are rich in loops and rich in beta sheets. So here is a review that just came out last year of all the de novo protein structures that exist in the protein data bank. And you can see they're highly enriched for just helical proteins, very rich in helices. And there's just a handful of them that actually have these beta sheets in them. And these beta sheets and generating these loops is very important. And one application that they're important is for like antibody design, right? So here is a protein I work with my Houston Methodist collaborator, Jimmy Gallagher, that we solved during the pandemic. This is a antibody that he found in one of the professor's arms here at UT and used it to actually study COVID and make an antibody therapeutic for COVID. And this is the spike, the receptor binding domain of the COVID-19. So this is the antigen that most therapeutics for COVID go after, trying to block. And you can see these proteins, including the COVID antibody antigen, is rich in these arrows, these beta sheets, and it's rich in these loops, these really long loops that don't have much structure and are very dynamic. And you can see in the antibodies themselves, they have no helices. They're purely beta proteins, and it's all about designing really dynamic and specific loops to actually be very specific, for example, targeting COVID versus targeting yourself, because that would lead to like an autoimmune disease. And David Baker actually followed this up with another diffusion model where they show that they actually have to fine-tune RF diffusion for antibodies. And even though they're trying to fine-tune them, you can see their error is still too high, right? They're still getting, above a 5 angstrom error. And these are some of the targets that they make. And in their paper, they show that for them to have success, they had to make over 9,000 different antibodies to find some that worked, that were pretty good binders. And when you think about it, making 9,000 de novo proteins, at a few $100s each, that's almost, $1 million in just DNA cost, without even doing any wet lab experiments afterwards. So it's still a work in progress. How do we use these diffusion models to make antibody therapeutics? But it was enough to raise $1 billion seed funding for their startup, so they're definitely going somewhere. The other thing is these proteins struggle with making these monomers or these single proteins that are longer. You can see the error kind of explodes as these proteins get longer. And you can see through nature the length of proteins based on if they're archaea, bacteria, eukaryotes. While like eukaryotes like us or like cats, animals, plants, fungus, their protein tends to get much longer. And you can see that once we kind of pass this 300 threshold, that things kind of start exploding. You can see that we're unable to do diffusion on a big part of protein space, everything to the right of this red line. So this is an area of active research of how do we scale these generative models to kind of make whatever proteins we need. And this is kind of what I get at when I'm talking about how do we use diffusion to cover the protein structure landscape. Because right now, we can't do this part of the landscape well at all. And then here is the motif scaffolding. Again, we have these motifs. We want to embed them in proteins. And they show some success, but even in this really small benchmark, a significant fraction of them, we still can't motif scaffold. So this is an area of active research. So that was the breakthrough model in this space. And this is kind of what the landscape looks like. There are models that are pure sequence-based diffusion, pure structure-based diffusion. New models are combining, doing joint diffusion on sequence and structure. And also, how do you do structure diffusion where you're conditioning on sequence? So the project that we've been working on, we actually started building off of Genie 2. So I'm going to briefly discuss what Genie 2 is. This was a diffusion model that just came out last year. And their big thing was they got really good at single motif scaffolding and show out of distribution generalization to multi-motif scaffolding, where you have multiple motifs and you want to generate one protein that has both of them, or 3 of them, or 4 of them. And their big innovation on the ML side was, instead of just training with a regular loss to do unconditional generation, they actually break the loss down into how well do you model the motif, which is what this M is, and on the left side of this equation, and how well do you model the rest of the protein, which we refer to as the scaffold, which is on the right side of this equation. But they actually never train it to do these multi-motif. This is just at inference time, they show that the model just generalizes to it, which is what their big thing was. So the other big thing that they had in their paper was, because of AlphaFold's big breakthrough in the last few years, there's this thing now called AlphaFold DB, which is they computationally folded over 200 million proteins. So can we use these 200 million protein structures to train diffusion models? And they were the first to do that. So for reference, RF diffusion was trained on the PDB with about 20,000 proteins. AlphaFold DB has about 214 million proteins. They were then able to cluster it down to about 2.3 million proteins. And then when you take the high confidence proteins in that subset, remember this PLDDT metric I talked about at the beginning, where they set the threshold at 80, they're able to get about 600,000 proteins to train, which is a huge increase in data from 20,000 to almost 600,000 proteins. So here's kind of how this clustering works to make this data set. I'm going to briefly talk over it because it's relevant for later parts of the talk. They essentially do some sequence clustering, identify all of these proteins, then they select the ones that are highest confidence, and then use this new structure clustering tool called FoldSeq to generate about 18 million proteins. A lot of these proteins are just small little fragments, so they filter them out. That gives them about 2.3 million. From those 2.3 million, about 31% of these clusters come from about 4% of the protein sequences. And this is very important because it really boosts the diversity, and we're not just sampling the same proteins we see over and over, but we enrich for this small subset of structure space that is not very abundant in our databases, but is very important for learning if you want to make a de novo generative diffusion model. The next thing, so this is a fact that we're going to find really interesting later in the talk, but you can essentially understand these proteins using a hierarchy. So like, is it primarily alpha or beta? That would be like the C level. And then you can just get more nuance, go to like the architecture level, the topology level. These three are purely looking at 3D geometries of the proteins, while the last one is looking at their evolutionary relationships. So interestingly, their clustering paradigm led to where each cluster, 97% of all proteins within a cluster all had the same H level. So they were all evolutionarily related, which is an interesting fact that we actually exploit in our model. So evaluating Gini 2. So again, here is RF diffusion's plot from earlier today, where the models kind of blow up as the proteins get longer. And you can see they don't make too much progress on scaling to longer proteins. Again, as you get longer, the designability just kind of plummets. But they do make significant progress in diversity. So at every length, they're actually able to make very different proteins. And again, this has to do with the fact that the data set's much, much larger, and you see a lot more diversity in structure space. And the model is also out of distribution a little bit because they only trained it up to 256 amino acids. So again, looking at how bacteria and eukaryotes protein distributions are, they're still unable to scale to everything to the right of this red line. Next, they're also unable to generate these beta sheet proteins. You can see that this is the data set for training, AFDB. It has a decent amount of beta sheets. Gini is very shifted over here to the right on these alpha helices, where the y-axis would be beta sheets. And on motif scaffolding, they do improve over RF diffusion, but it still needs more work. So the next thing to look at is whenever you sample, how much noise do you add in the diffusion process? Which is what we call temperature. So high temperature is high noise, low temperature is low noise. You can see that as they lower the temperature, they're actually able to improve the diversity and designability of these models. And that's actually what enabled them to make this motif scaffolding improvements. But as you lower the noise sampling or the temperature, you can see you get secondary structure collapse, where they're just only generating helical proteins, and they lose their ability to generate beta sheet proteins. And then this is our segue into the progress that we've been doing here at IFML. So here's a 1-minute clip from the author of Gini.
More challenging to work with because of the quality of it. So I think in one of the figures, it shows that if we're using our own designability metric using the SCRMSD, only like 40% something are actually designable. But if we're using the SCTN, around 80% of them are designable. So essentially, structures in the AFDB so to us, it feels like the structure is pretty much correct, but there's a lot of refinement that need to be done in terms of at least locally. So it would be hard to say that is it a fair comparison? It is biased or not in some sense. Like we have more data, but the data is much more noisy.
So,
in some sense. So there's no clear answer here.
So at the very end of that, he said something very important. And he said that we have more data in AlphaFold DB, right? They had 214 million proteins compared to 20,000. But the data is much, much more noisy. And that manifests itself here, where if you use root mean square deviation or this TM metric, which is how well you can align proteins in 3D space, you can see that even though the PDB, which is real experimental data, is much, much smaller, it's actually a higher quality data set compared to AlphaFold DB. And this is kind of where our project steps in, because, and this is where ambient protein diffusion comes in. And ambient protein diffusion is learning from noisy protein structures in AlphaFold DB. So to go a high level over what ambient diffusion is, we have a great collaborator, Yannis Daras. He did his PhD, graduated here from UT in November, where he invented ambient diffusion. And by ambient diffusion, I mean like just whiteboards of just inventing new math. And just, it was actually really beautiful to see him do it. But essentially the way it works is we have clean data. So taking a step back, right? We have this diffusion where we slowly add noise and then we move the data along. So 0 would be 0 amount of noise, and then T would be where it's 100% noise. And ideally, whenever we're doing diffusion, we try to use only clean data. But so we throw away 99% of our data because it's dirty for some reason. So what he came up with is, can we generate this, there's usually natural amounts, he calls it T nature, natural amounts of noise in the real world data. So how do we find where this T nature is, use everything to the right of this wall as clean data, and everything to the left of this wall as noisy data. So like right now, all of this is thrown away. How do we find a way to actually use it without corrupting the model, right? We don't want the model, if an image is, to start diffusing blurry images or noisy images. We want the model to generate beautiful, high resolution images, but can we still learn something if we train with blurry images? So how do we do this in proteins? And this is what ambient diffusion is. He came up with a way of training the model where we don't have access to T0. We only have real world data. So we only have images or proteins at T nature. So how do we do some math tricks to train the model to learn how to generate T0, even though it's never seen it during training? And that's what this equation that he came up with here enables, where the model outputs all the extra noise to get you to T nature, and then with some more math, we can remove all the noise and end up at T0. So we're gonna apply this ambient diffusion technique to proteins. So again, just going through that again in proteins, right now Genie is trained where all proteins have a PLDDT greater than 80. How do we make use of this data? So you can see a lot of the data, most of the distribution shown here in orange is actually below 80. So we're throwing away most of our data. How do we actually make use of the data here with ambient diffusion? So here's an example of what low quality protein data would look like. So before we were looking at images, it would look kind of like this. So what do these kind of images look like in the protein world? Right, so you can see this is well folded, this is well folded, but here's all this noisy, where AlphaFold was unable to fold the actual protein. And I call this, we just have noodles, little noodles dangling off, which means AlphaFold doesn't know where to put the atoms. It's just so, but it knows that they're connected in a string, so it looks like a noodle, but we don't know where they belong in 3D space. So this can corrupt the model while training, because it's just gonna learn to generate these noodles, which are not useful in any downstream application. But this part, this well-folded, these are called little domains, is high quality, very useful data. So how do we learn from this and ignore the noodles with ambient diffusion? So while we were like figuring out how to use ambient diffusion, looking at the data, looking at the quality of the data, the other thing we noticed is this data set where they clustered AlphaFold DB into 2.3 million proteins, and it was supposed to be all that in a bag of chips, actually was filled with duplicates. So for example, here's an example where when we actually clustered the data ourselves, and you can see here, does anybody want to guess how many proteins this is?
How many proteins?
Yeah, how many proteins did I superimpose here?
At least 2.
There's 4 proteins here, right? Here, there's 2 proteins, but you can see they're like near duplicates, and where they differ is at their noodles, right? You can see this noodle zigged, that noodle zagged, right? Which is irrelevant to training the model, right? Like this is just noise, right? We don't want to be thinking these are 2 different proteins when they're really identical at wherever the model was accurate, and just the difference is just noise. So we cleaned up the data set, and then we applied it, and then we trained with ambient diffusion. So here is a more deeper look at our cleaning of the data. So you can see before they were using almost 600,000 for Genie 2, and this is using a PLDDT threshold of 80. So they went from 2.3 million to about 600,000 using that threshold. But after our deduplication, we were able to bring it down by 12x at this threshold, or at 6x nearly, and for the larger data set, 16x and 8x. So there's large amounts of duplicates in the current data set the whole field is using. So let me kind of show you now what ambient protein diffusion can do. So here is kind of this plot where we're looking at the trade-off between designability and diversity, where diversity is on the x-axis and designability is on the y-axis. The best models would be on this top right-hand corner, where you have the best, everything that it generates is designable, and it's super diverse. And you can see here is kind of all the models that exist, and you want to be on this outer edge. We call this the Pareto frontier. And you can see here is RF diffusion, and you can see Genie made some significant progress by moving to this upper right-hand corner. But here's ambient diffusion, right? So we train models varying different parameters, data set size, the amount of noise you use in ambient diffusion. And you can see our model is along the entire outer edge, with these two models being the best compromise between designability and diversity. For example, you can see here is RF diffusion, here is Genie, and you can see we increase our diversity significantly while having marginal change in designability. And then here is again now comparing ambient protein diffusion for longer proteins. So here in blue is Genie, which is the model and the code base we started working on to develop ambient diffusion. And then here's whenever we train it, whenever we just train it using the exact same method as Genie, but using ambient diffusion, you can see our model improves significantly. And then whenever we actually fine tune it or show it more data with longer proteins, you can see it gets much, much better at longer proteins. So while we've been developing this over the last few months, NVIDIA actually started making generative protein models. And this is a tweet from a guy at NVIDIA from last night. So I think he tweeted this yesterday. I took the screenshot last night, where they just came up with this new model called Proteina, which is just protein in Spanish. And what they show is they just did what NVIDIA could do compared to us academics. They just went big, right? So they made a 400 million parameter model, which is an order of magnitude bigger than our model. And they, instead of using the 2.3 million proteins that we had from that paper, they grabbed all the proteins that are nearest neighbors of it to make it 21 million proteins. And so they went big on data and went big on model. And when they did this, you can see they're just crushing everybody when it comes to longer proteins, which makes sense. They had more data and they scaled to longer proteins, where they were training up to 800 amino acid proteins. And they improved motif scaffolding. So here's Proteina versus Genie. They made improvements on this end. But here's ambient diffusion versus ambient protein diffusion versus Proteina. So you can see our model is competitive on designability, but we're actually crushing them on diversity by our ability to go into these noisy proteins. So it feels pretty good to see that, we have a model that is an order of magnitude smaller than theirs and is very, very competitive. And it's trained with several orders of magnitude less data, right? We didn't use 21 million proteins to train it. So it's a work in progress. We're still figuring out the motif scaffolding, the secondary structure evaluation, and developing new metrics for other applications in protein diffusion. And our goal is here in the short term to start actually making proteins in the lab and be the first to show that we can indeed scale protein diffusion to 500, 600 amino acid proteins, which has never been done before. I'd like to thank our team, Adam, Jeff, Giannis, and me. I'll take any questions. Thank you.
Thanks. Great talk, Danny. Any questions for Danny?
You mentioned the early on trouble in sort of the secondary to tertiary structure. Is some of that in the linkages, like the disulfide linkages, or is that part solved?
Yeah, so this is backbone diffusion. So there are no side chain atoms. So the model doesn't know what a disulfide bridge is.
Right, okay, makes sense.
Mm-hmm. However, whenever we forward fold some of those sequences that we generate for our backbones, we have observed that we generate disulfide bridges in the all atom versions.
So I was wondering, what will enable your research further? What are your limitations computationally? Like, could you apply the NVIDIA machine learning? Would that enhance what you guys are doing, even though you have a smaller data set? Are you limited on data? And also, the second question is, how much sharing is going on within the community? Are you guys sharing very broadly the data, for instance, and the methods and techniques?
Yeah, so that's so how do I answer that? So
We open source everything.
yeah. So NVIDIA just open sourced their code last night. We were aware of this model, I think, sometime in January. And we were happy to see that we were already like competitive on designability and beating them on diversity. We've made a lot of improvements since then, and we're currently working on combining them to our latest model. So that plot I showed there is like from a model from like 3 or 4 weeks ago. Now, when it comes to like limitations, Center for Generative AI has been great. We've been able to actually compete because of Center of Generative AI. So to train one of these models, a normal run takes 10 to 20 hours, depending on a few parameters, on 48 H100s. So that's 48 different computers training one model. And we're able to do that here at Center for Generative AI. However, if I'm running 1 or 2 models that are 48 GPUs, Giannis is running 1 or 2 models at 48 GPUs, and Jeff's running 1 or 2 models, and we're running all these experiments in parallel, you know, we're using up most of the cluster, right? 2/3, 3/4 of the cluster. So we're kind of hogging all the GPUs. So if we had an extra, 500, 1,000 GPUs, that would be great. So we're always compute limited. I know Adam's working on that. But yeah, so, and then on the data side, it's not very clear because we made a lot of our improvements by deduplicating data, right? Like one of our models that's in the Pareto frontier, I think was trained with 50,000 proteins compared to like 600,000 or 2.3 million or 21 million. Like, and it's one of the state of the art. So it's being smarter with the data, and to improve on that end, what we need is just like more like technical manpower, right? Just having more people to run experiments. We have different ideas, validate them, having more people who can work at the interface of proteins and machine learning and data engineering to see what works and what doesn't work. That's really our band.
But I think it's an interesting open question, and one we're trying to address in this project is, since the early 20th century when people were trying to do X-ray crystallography, there's always been this issue of, how much signal is in your structure, how clean is your structure, what's the resolution of your structure. Mm-hmm. That noise is introduced in many, many ways. And what is the best way of extracting all of the signal from a particular structure, both computationally or generated in the wet lab? Ambient diffusion is an attempt at that, but for sure there could be better ways, and we're actively thinking about that. Question in the back. I'll come to you in a second. Go ahead.
This is a higher level question, but given the magnitude of resources and compute that big corporate has now, how do you see academic work dovetailing with what they're doing, given the, this NVIDIA situation? Yeah,
so one thing I've learned over the last few years is that compute is a double-edged sword. It's really nice because you can run more experiments, you can scale, things like Proteina, but it makes you very mentally lazy. You just like throw more compute at it, throw more money at it, throw more hours at it, throw more data at it. And we've had to actually be very clever and come up with algorithms and data tricks and a variety of other techniques to really squeeze what we can out of it. So I think it's actually, I think us not having NVIDIA's compute for this project is what enables us to invent, you know, ambient diffusion for proteins, all of our data engineering tricks.
That said, we would take more, you know, GPUs if we had them. But yeah, I think that.
Director of Center of Gen AI, please give us more.
Yeah, exactly.
Hi, thank you very much for this wonderful talk. You've talked about single proteins, homo multimers. We are working extensively with HLA complexes, which are composed of 3 different units, right? 35 kilodalton protein, a small 10, 12 kilodalton protein, and a peptide. So on one hand, this is very fascinating and very interesting moving forward also for immunotherapy applications, cancer vaccine development, and so on. But at the other side, I'm wondering where you are in terms of predicting the structures of these very diverse complexes, which are very similar to each other, thousands of different variants, which are 1, 2, 3 amino acids apart from each other, but very important, of course, then in this diversity in immunological responses.
Yeah, so these HLA genes, this is for like MHCs, right? And like is the VDJ recombinations? But it's for the MHC complexes. So that's more of structure prediction. So that's like an orthogonal field of what we're working on here. Very important, but like how do you actually model these like small amino acid changes between like alleles or like people in the population? That's just a different field as a whole.
Well, that said, I mean, we've worked a little bit about that on active sites and enzymes. You know, if you happen to tweak one particular place right in the active site, you need your model to be super, super responsive.
Yeah, but what he's talking about is more of like immunotherapy, immunology, which is like a very like structure poor area.
I see.
And like gene, like we just don't have like the data to like really fine tune models in that space really well. Like we don't have like, you know, thousands or tens of thousands of HLA structure complexes, you know, crystallized to even train a model on that. So it's hard to like, how do you take bacterial proteins and bacterial enzymes and everything else in the PDB and try to like predict that niche application? It's a very data poor region, at least currently.
Other questions? Okay, we'll take one last follow-up here.
Again, thank you for this. It's amazing. I was just curious, in other fields doing ML, they're using synthetic data. And I was wondering if that's something that's applicable to what you guys are doing.
Yeah, so that was one of the main things that we actually did here, because we used the AlphaFold database, which is primarily synthetic data. It's all just like another AI model predicted this data, which is how we were able to have 214 million protein structures to begin clustering from. But a lot of that data, it's relatively low quality, right? Most of it comes from like genome sequencing of like dirt or like the ocean or like some random leaf that somebody walked by, and they just like sequence the DNA on it. And then using like the rules of biology and kind of what's known, they're like, okay, we believe a protein begins at this position in the DNA and it ends at this position in the DNA, but nobody actually went in there and confirmed, right? It's just like putative annotations, and then people at Google took these putative annotations from these metagenomic sequences and all this sequencing data, and they started just folding them into proteins, hoping that they're real. So I'm not saying most of them aren't real, just saying we don't know. It's all synthetic data in a way.
Okay, let's thank Danny again.